Hello friends, welcome back to my channel Plant Pathology Pathsala. So as you all know that we are discussing the isolation and purification of plant viruses. So in the first part we have already covered the isolation or extraction part. Then we have already discussed the isolation and purification. What is the need of this? Then the purpose, principle, about the steps followed in the extraction. So in the extraction we got this crude virus extract which then will be subjected for the purification. And the extraction buffer component we have discussed all about its function, why it is required, everything we have covered with precautions. So in the today's video, the part 2, we will discuss the virus purification process. So without delay, let's start the video. So virus purification. So objective of purification is to isolate virus from infected plant tissue. Yeah. Then the major thing, after extraction, what we got? We got the crude virus extract. So the host contaminant need to be removed. And why we are doing the purification? To obtain highly purified virus particles. So this virus purification is actually contain three major steps, that are clarification, partial purification and final purification. So total the isolation as well as the extraction and purification of plant viruses include the extraction, clarification, partial purification, final purification, and ultimately we will get the finally purified virus. So let's discuss all these steps in detail. First one is clarification of the virus extract. So what is the purpose? So the first purpose of this clarification is to remove the coarse plant debris. So before clarification or after the extraction of plant virus from the infected samples, we got this type of crude extract. So it contains both the plant debris as well as various chloroplasts, starch granules, many contaminants are there. So by the clarification steps, we remove the coarse plant debris, and ultimately after clarification we got this clear virus containing extract, from crude plant extract to clear virus containing extract. So principle behind the clarification is the virus particles remain suspended in the solution of heavy plant debris that is removed by low speed centrifugation. Okay. So how this crude virus extract is changed to this clear virus containing extract? By low speed centrifugation. Okay. And also we will use some chemicals that will for the maintaining the stability as well as the integrity as well as infectivity of the plant virus. So outcome will be the clear virus containing extract without coarse plant debris. Okay. So this is the basic about the clarification step. So then what are the chemicals used with their function in clarification process? So generally in the clarification process we use organic solvents. So what this organic solvent does? So this organic solvent actually dissolves the membrane and lipids, and lipids of the plant. Okay, so then the host protein will coagulate, and when it is subjected to the low speed centrifugation, we will get the clear virus containing extract on the top layer. Okay, in the middle layer there will be the coagulated protein or cellular debris, and in the bottom the organic phase, that the organic solvent we have used with some pigments, lipid or pigments it will contain. Okay, three phases we will found, and we will take the supernatant, that is the top layer that is containing the virus particles. So the solvent or organic solvent that are mostly used for the clarification of the virus extract are, first is most important is the chloroform. So what it does, it actually remove the lipid as well as membrane protein. I have already discussed this. Okay, so it denatures the unwanted host protein and helps in separation of plant debris. So similar thing also done by this butanol, and butanol it also removes the chlorophyll, but this butanol is actually more efficient than the chloroform. It is less harsh and preserve virus particle better than the chloroform. Okay, remember this compound, these two are very important. And phosphate buffer means it will maintain the pH osmotic balance and ultimately preserve the virus stability as well as virus infectivity. Okay, so this is about the chemicals used in clarification process. Okay, next, the second step, that is partial purification. Here three steps I have discussed, means I have told you in there are three major steps in purification: the clarification, partial purification, and final purification. So clarification means we will remove the coarse plant debris or chlorophyll or starch granules. This major portion, that coarse material, we will remove by low speed centrifugation and using some organic solvents, and ultimately we will get the clear virus containing extract. But still this contain some soluble proteins, lipids, or some soluble compounds. It is not the pure virus. So to get pure virus, it will be further subjected to partial purification and final purification. So first the partial purification process. So after clarification, the extract still contain soluble impurities that must be removed. So main purpose is to remove the soluble contaminants while preserving the viral infectivity. So what are that component that need to be, or contaminant that need to be removed? That are host proteins, metal ions, nucleic acid of the plant, enzyme, phenolic compound, and other soluble impurities. So the major principle is specific chemical and reagent are added to the virus, means the extract, so that will protect the virus and also remove the soluble contaminant from the extract. So what are the chemicals that are used? We have to use some reducing agent like metamercaptoethanol, sodium sulfide. Also chelating agent also we have to use like EDTA, antioxidant, then polyvinyl, pyrrolidone. So these are some chemical groups that generally we use during the partial purification process. Here also same thing like the clarification process. After adding the reagents, these type of reagents, it is subjected to low speed centrifugation. So in both the clarification as well as partial purification process, after adding the reagent, the extract will be subjected to low speed centrifugation and we will take the supernatant. Okay, in the clarification step we use the organic solvent, but in case of partial purification we use reducing agent, chelating agents, antioxidant, PVP. So many type of chemicals are added to retain the virus infectivity as well as to remove the soluble impurities. So let's discuss the chemicals or reagents used in partial purification step with their function. So first about the reducing agent. Reducing agent, I have already told that reducing agent actually prevent the oxidation or browning, okay, thereby protect the viral proteins and maintain the infectivity during the purification process. So what are the strong reagents or reducing agents that are used? Very important is the beta mercaptoethanol. This is the most important reducing or most commonly reducing agent used in virus purification. What is how it does its role? So it breaks the disulfide bond, it prevents oxidation of phenolic compounds, protect the viral coat protein, maintain SH group in reduced state, and improves the viral infectivity and recovery. So actually what the reducing agent does, it prevent the precipitation of proteins, coagulation of protein, and also the prevention of oxidation. Okay. So what is the concentration generally used? The concentration is 0.1 to 2% volume by volume in extraction buffer. Then the second most important reducing agent is dithiotheritol DTT. It's also a strong reducing agent. It also maintains the sulfhydryl group, prevent protein aggregation. So major thing that the reducing agent does is it prevent the protein aggregation or protein coagulation. Similarly, the sodium sulfide also, it's also a reducing agent that prevent the oxidation as well as protein coagulation. Okay, this is about the reducing agent. Next, about the antioxidant. So antioxidant means it prevents oxidation. So examples of commonly used antioxidants are ascorbic acid, sodium sulfide, cysteine. So it prevents oxidation of phenolic compounds, prevent browning, and maintain infectivity and stability of the virus. Next is the EDTA or the chelating agent. This is the most very important chemical. So how it prevent the or protect the plant virus? So it is not acting as any contaminant removal chemical. It is a virus protectant. How? Because the EDTA act as chelating agent. It chelates the metal ion. By chelation of metal ion, it inhibits various enzyme, degrading enzyme like proteases, nucleases, because virus is a nucleoprotein structure. So during extraction, if plant from the plant cell, if nucleases or proteases are released, it may degrade the viral protein as well as viral nucleic acid. So by chelation, this EDTA inhibit the metal ion to act with along with the nucleases as well as the proteases, because the enzymes, the nucleases or proteases require a metal ion, that is the cofactor, to become active. Without metal ion, it become inactive. Okay. This is about the chelating agent. Then about the PVP. PVP, it polyvinyl pyrrolidine. It actually helpful in the removal of the phenolic compounds. It binds to the phenolic compound, prevent the quinone formation, and it removes the phenolics by centrifugation. So it acts as the act in the purification steps, whereas this EDTA as act in the maintaining the virus stability as well as infectivity. So this is about the chemicals used in partial purification step. Then final step is final purification of plant virus. Three steps we have discussed: clarification, partial purification, then ultimately the final purification of plant viruses. Major objective of this is to obtain highly pure concentrated virus particle suitable for downstream application. What downstream application? Like identification, characterization, physiological studies as well as infectivity studies of virus. So objective is to get purified virus without any type of contaminants, both soluble as well as insoluble. Then to preserve virus infectivity as well as the structural integrity. So methods used are differential centrifugation, ultracentrifugation, density gradient centrifugation, isoelectric precipitation method, salt precipitation method. So various methods are there, 5 methods. We will discuss all this one by one. Why it is important? Because it provides pure virus for research. For because if you will not get the pure virus, you can do the further work. You can perform the identification study or infectivity study that can be used in electron microscopy, ELISA, genome extraction, sequencing. So for various downstream applications, the purified virus is needed for the research especially. So this final purification is very essential. So what are the methods used in final purification? The first one is the differential centrifugation. So here the particles are separated based on size and mass. So the supernatant we have collected after the partial purification process, that will be subjected to high speed centrifugation at 80,000 to 1,20,000. Then virus pellet we will get that after centrifugation, that will be suspended in the any buffer. The buffer may be mostly generally the phosphate buffer. Okay. uh, this is about the differential and density gradient. So the major difference between density gradient and the differential centrifugation is, in the density gradient the particle actually migrate to region where their density is equal to density of the medium. So a specific medium is used, either sucrose or cesium chloride. Against that medium, the particle will migrate. Okay, but here no such gradient is used. Particles are generally separated based on size and mass. So this is about the differential centrifugation, separation based on size and the mass. Based on size and mass. And in differential centrifugation, increasing centrifugal force remove larger debris first, then the pellet the virus particle at high speed. So major principle behind this is, principle behind the differential centrifugation is particle sediment at different rate depending on their size, shape and mass, just like other centrifugation process. This is very common. Purpose is to remove the debris, nucleic acid, chloroplast, whatever left behind, and we will get ultimately the purified virus. Okay, so same process discussed here, given here, the clarified one that will be subjected to low speed centrifugation. Then this pellet we will get, then again it will be subjected to high speed centrifugation in differential centrifugation, and ultimately the purified virus pellet we will get, that will be suspended in stable buffer like the phosphate buffer. Next is the density gradient centrifugation. As I have already told, here the particles are separated in gradient medium according to their density. So that gradient medium generally used are sucrose gradient as well as cesium chloride gradient. Here the centrifugation speed is high, that is 50 to 70 gram. Then mostly used for rod shaped virus. Okay, and density gradient is generally mostly used purification method. Advantages: it concentrates virus without pelleting and gives high purity, because pelleting, after pelleting it may create some difficulties for again suspending inside the sample, that buffer. So without pelleting, it concentrates the virus particles and gives high purity. So after the partial purification, whatever the clarified sample we will get, that will be subjected to high speed centrifugation of 50 to 70,000. Then we will get distinct bands and virus will be collected carefully. So two gradient here we have used sucrose as well as cesium chloride. Next, other purification methods are ultracentrifugation, then salt precipitation method, and last one is the isoelectric precipitation. So in the ultracentrifugation, the principle is the virus is subjected to high speed centrifugal force, that is 40 to 80,000 for 1 to 3 hours. Okay, whereas in salt precipitation, salt is used. So virus is precipitated by addition of salt that reduce its solubility, and ultimately virus will be precipitated in the bottom. So generally this ammonium sulfate 25% is mostly used. Okay, and here low speed centrifugation are used, 3000 to 5000 for 15 minutes only. So the with that partial purified virus extract, 25% ammonium sulfate will be added and it will be subjected to low speed centrifugation, 3000 to 5000 G for 15 minutes, and we will get, means that virus will be precipitated by the help of the salt, and ultimately we will get the pellet, the virus particles, which will be suspended in the stable buffer or phosphate buffer. But the major thing we have to keep in mind is, see, viruses are inactivated by salts. So it can be used for the viruses that are not inactivated by the salt solution. So this is not mostly used method. Generally centrifugation methods, especially the density gradient centrifugation method, is most commonly used for virus purification. Next method is isoelectric point. So here the virus is precipitated by pH change. So major principle is, the virus actually, it's an amphoteric molecule, both positive as well as negative molecules are there. Positive charge imparted by the proteins and the negative charge by the nucleic acid. So it's an amphoteric molecule. So what we does, we generally do acidification. See, at isoelectric point, the net charge will be 0. So by this 0 charge, it will, the virus electrostatic repulsion between the opposite charges will be minimized and virus become aggregated and precipitate at isoelectric point. And that isoelectric point for most of the virus ranges from this 4 to 5. Okay, mostly less than 4.8. So we have to do the acidification of the virus extract. So if we acidify the extract, then ultimately that acidification will help in maintaining the isoelectric point, and ultimately the virus will be aggregated or precipitated in the bottom. Okay, this is the key principle behind the isoelectric precipitation method for the virus purification. Okay, generally this, if in the MCQ based question if it is asked, then what is the isoelectric point for the virus precipitation, that is less than 4.5 or more specifically 4.8 plus minus 0.5. So this is about the methods of final viral purification, that is the density gradient centrifugation, then ultracentrifugation, salt precipitation, and isoelectric precipitation. Then ultimately we will get the virus purified form, in purified form. How to check the virus concentration as well as its purity? We will check that using spectrophotometer. So colorimetrically we can use, we have to estimate, that is by using the nanospectrophotometer. I have already discussed this, that in the previous video about virus diagnosis, I think. virus diagnosis methods, I think. In that video, I have already told that the nucleic acid absorbed the UV light at 280 to 60 nm, whereas protein absorb it at 20 to 80 nm, and this absorbance ratio will indicate the virus purity. This is very important, 260 by 280 ratio. So this ratio indicate the contamination as well as purity. If it is ranges from 1 to 1.2, it indicate the protein contamination is high. If it is 1.2 to 1.8, we can say virus is moderately pure. If it is more about 1.8 to 2.2, the virus is highly pure. So it is for the estimation of virus concentration as well as the virus purity. Then we have to store the virus. So storage of virus extract will be done either at minus 80 degree using few drops of sodium azide 2%. Why this sodium azide is used? To prevent the microbial growth. This is very important. Okay, sodium azide 2%. Then we can store it in lyophilized form, means freezing dried. And the virus suspension will be freezed, then vacuum drying will be done, and it can be stored at either 4 degree or minus 20 degree for long-term storage. Apart from this, you can store the virus in liquid nitrogen by adding equal amount of glycerol. Okay, so this is about the storage method. So in summary, here we have discussed three major steps in virus purification, that are clarification, here the coarse debris will be removed, then partial purification. In that step, soluble contaminants will be removed, and the final purification to get highly pure and concentrated virus particles that will be then subjected for virus concentration as well as purity estimation using nanospectrophotometer, that is by using 260 by 280 ratio, and then it will be stored either by using lyophilized form or in liquid nitrogen or at minus 20 degree with 2% sodium azide. So this is all about virus isolation as well as purification and storage. So I hope now the virus isolation purification process is very much clear to you people. So thank you for watching this video.